In this video, we will discuss about some of the basic principles of chemotherapy in brief and how it can be used to treat the cancers. So, the basic principle of chemotherapy is that it will induce cellular stress on the cellular stress near the tumor and suppress the cell defense system. This tumor has some defense systems such as tumor microenvironment that is usually found in the solid tumors. It will suppress those tumor cell defense system, and then it will initiate apoptosis on those cells that will lead to the cancer, that will lead to the killing of the cells, and most of them are cancerous cells. And this is how it will works in the graphical image, that there are cancerous cells surrounded by the healthy cells. When we inject the drugs, most of these cells will uh, will be will most of these cells induce apoptosis or programmed cell death, and during this step the some of the healthy cells will also get destroyed or killed. And the goal is that complete remission. We have to get rid of all the tumor cells, and that will lead to the prolonged life, and it will avoid, since if all the tumor cells are eliminated, then it will avoid reoccurrence and provide comfort. So what are the basic stages of chemotherapy drugs? Is that it can be induced at different cell cycle stage. First one is this, the non-specific drugs that will it will have non-specific drugs will mainly work on this at G2 stage, where it will work towards this DNA repair or DNA checking, as most of the tumor cells has this DNA damage properties. And some of these drugs are commonly known as tumor antibiotics, alkylating agent, platinum agent. And in the M phase, the target of this chemotherapy drugs is on this cytokinesis, that is prevent the cytokinesis from the tumor cells that uh, that usually supports the tumor cells proliferation and helps the tumor cells in its defense system, such as tumor micro such as improvising tumor microenvironment. So if cytokinesis will be targeted, then cell's defense system will get disrupted. The other things that can be affected is during this G1 phase and G0 phase is this antineoplastic enzymes, and these antineoplastic enzymes are usually responsible for proliferation and uncontrolled growth rate of the cells. And by targeting this antineoplastic enzyme, it can trigger apoptosis. And finally, in this the replication step, they can focus on this topoisomerase inhibitors. We have discussed in our previous videos that what is the role of the topoisomerase in the most of the immortal cell lines. This topoisomerase has been mutated in a way that it will not get degenerated with the cell replication. However, in the primary cell lines, this topoisomerase will get disrupted as the cells replicates its cells, and because of this, when the topoisomerase completely degenerated from the cells, leads to the it will leads to the uh, it will leads to the inability of the cells to further replicate, and that is the reason the primary cell lines has the capability to replicate just for like 7 to 8 times. So in the S phase, the target of this chemotherapeutic drugs is on this topoisomerase inhibitor. It will inhibit the topoisomerase so that cells will not be able to replicate itself. And how this chemotherapy or immunotherapeutic drug works is that the they will the tumor cells has some non-specific, or tumor cells has some specific antigens that are unique to the tumor cells. It will be detected by antigen presenting cells, and that it will provide those antigen information to the CD4 plus T cells, and it will activate the T cells. And this activated T cells will provide these informations to the genetics. The activated T cells will not this CD8 plus T cells, activated T cells will get information also from these activated dendritic cells, and thereby these T cells now will be able to generate antibodies against those specific antigens and can target the immune cells. And these are some of the examples that can be used. So, and these some of these examples are related to CAR T cells also. So in this case, what happens? The drug is targeted drug delivery. In this, they will drug will have the information about the specific antigens that are present on the tumor cells, so that it can target the tumor cells but not the normal cells. That can be seen here. The cancer cell specific surface receptors will be detected by these drug chemicals that can interact with those cancer cell specific surface receptors, and in this case the normal cells will not get affected, as we can see that normal cells will not be able to interact with this drug. And in the second case, where personalized medicine can be used, in this case the mutations in the DNA of a patient will be analyzed using different omics strategies such as genomic transcriptomics, epigenetomics, proteomics, phosphoproteomics, and metabolomics. And after mutation is identified, the drug will be made that directly interact with the mutations that are found within the patients. And those drugs since it is customized to the patients, it will have higher effectiveness compared to the case where it is injected with the generic drugs. And next thing is about the immunotherapy. It is related to the CAR T cell therapy. In this case, patient's blood is sample is taken, and then they will isolate the T cells and reprogram the T cells to target the cancer cells, or we can say they will inject the chimeric antigen receptors on these T cells, and then apply and then again transduce those T cells into the patients. And this is when it is transferred to the patient, the T cells will be able to interact with the cancer cells, since now they have informations about the cancer specific antigens. So in this case, the T cells activation time reduced, and it can activated T cells can directly target the cancer cells. These are just a basic principle about chemotherapy and how it can be used to in to treat the patients who are affected with the cancer without too much impact on the healthy cells, or without too much side effects. However, these things are still under studies before however these things are still under we can say ongoing. How the research is still ongoing towards the improvement of these processes. If you need to understand more about this chemotherapy and how their chemotherapy idea can be applied to the patients so that they will have minimum side effects, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.